One, welcome to Lab 10. In Lab 10, we're going to discuss the urinary system. Now, of course, this being lab, we're going to focus on anatomy. The thing I always like to start almost every single chapter with, at least some of the new recordings out there, my new notes, and will have the quotes. My older notes don't, so you guys wouldn't see these in every single lab recording that I've done right now. But I chose one from Charles Darwin that says, "A man who dares to waste 1 hour of time has not discovered the value of life." And of course, I chose it because it has waste, and I could not resist the pun because we're in the urinary system. Um, so kind of get that double pun in there. One of the things I talked about in lecture when I introduced the urinary system, you guys may remember that I said that the urinary system makes a wet bed. And if you're wondering what that means, I'm going to go over here to the actual lecture notes. If they will actually, there we go. I couldn't tell which one was which but I said it makes a wet bed. Is acid base balance, water volume control, electrolyte levels are controlled, toxins are removed, blood pressure is controlled, erythropoiesis is done, vitamin D and glucose is produced. So as we discuss this is what we basically were saying here, just in a slight different way. As I said, that we secrete various wastes, things like uric acid, urea, stuff like that, that we've discussed before, creatine, those kinds of waste products. Erythropoiesis, because it makes the hormone erythropoietin, EPO, or erythropoiesis stimulating hormone. Remember, that stimulates the production of red blood cells. We control pH. How do we control the pH of blood? Well, we can secrete or reabsorb hydrogen ions, or we can secrete and reabsorb bicarbonate ions at various parts of the nephron. And you guys saw that in lecture. I'll talk about that there. We will talk about the nephron here, but we're not going to go into the same levels of detail. Another thing we said was we control blood pressure. We make renin. Renin is secreted by the juxtaglomerular cells of the kidney we talked about in lecture. And one of the things that I wanted to make sure is we understood is it converted angiotensinogen to angiotensin 1, and that chemical reaction happens because of renin. It's participating in the synthesis of vitamin D. Remember that you have this molecule where that is made in the skin when UV light strikes cholesterol. That goes to the liver into an intermediate that goes to the kidney. Kidney takes that and converts it to vitamin D. Vitamin D3, which we know is involved in the uptake of calcium and phosphorus, phosphate ions for phosphorus. It's also involved in glucogenesis, sometimes called neoglucogenesis. This is when you make sugar molecules. What you can do is you can take molecules that come to the kidney, get there, and biochemically rearrange them into glucose, especially when you're starving. So it does glucogenesis during starvation. Now, we are going to see that in this anatomy, we're going to see there are two kidneys, two ureters, a single bladder, and a urethra. Now, as we go into this, I have some models I'm going to pull out. I couldn't do digestive models because of how hard they are to get that number of models here. They just weren't comfortable taking them out of the laboratory. But here I've got the urinary models I use. And as we go through these, we'll look at them. And I forgot that I did put this in here, my mnemonic device for remembering the functions here as well, that it makes a wet bed. But I always would pull that up when I was in class from my notes because sometimes we had not yet done it in lecture. Specifically in the summertime, I was primarily teaching this lab previous to the lecture. We would do lab, then we would do lecture. And then some semesters it would be lab, lecture, lab, depending on how big the class was for the summer. So there's that mnemonic, and it's there, but I did pull it off because I honestly forgot that it was in there. So we will see that we have two kidneys, the two ureters coming down from the kidneys, going to a single urinary bladder that holds that urine out the urethra. And these are the major organs of the urinary system whose job it is to take urine and get it out, the waste products out of the body. Now, as we talk about it, I am going to pull a model over here. The first model I'm actually going to use. Now, I will use a model in a minute that I do talk about. I will use. Um, and what I wanted to do is let me get this under the camera really good. I may need to move it just a little bit, make sure it's under the camera, right? This will be the model I'll be using, and I always like having the ability to bring these home and video them as well. The camera that the college has let me use is quite nice. So what I always like to do, you guys know that when I teach anatomy, the one thing I'm always going to do is tell us what we're going to look at and describe it first before we look at it. Um, to me, remember, as you approach anatomy, it's always best to know somewhat something about it before you look for it. Um, and that's the way I approach it. So as we go through, let's start with our renal capsule. Now, as this name applies, renal is associated to the kidney. Capsule, it surrounds and covers the outer surface. Kind of like the capsule that we have capsules like capsules in medicines, like a pill, a capsule pill. The capsule's the outer covering. So there's a layer of collagen fibers, and it's the outer surface of the kidney. Then underneath that, there is a superficial portion called the renal cortex, a superficial portion of kidney. Then a little deeper is going to be the renal medulla. Now, those are the three major layers: capsule, cortex, and medulla. Now, the pyramids are in the medulla. They are structures found in the medulla. Now, we know that a pyramid is usually built out of, it's a three-dimensional structure made out of triangles. So we're going to be thinking here this is a triangular structure, okay? And at the tip of each pyramid, we are going to find the renal papilla. That's the tip of the pyramid. Now, in this case, the base, the broad part of the pyramid's on the outside, where as we go inside, the pointy parts of the pyramid point inwards. Now, the renal column, we're going to see that's bands of tissue between pyramids. You have two pyramids side by side, and there will be a column between them. And I'll help you see this. The renal lobe, this is going to be if I were to go find one pyramid, find a pyramid, and then we go on top of it and on both sides of it. That's going to be a renal lobe. We have a renal pyramid, any of its overlying renal cortex, half of a renal column on one side, half a renal column on the other side. Now, there also is going to be the renal sinuses, and these are all the internal cavities inside the kidney, which some of these internal things have associated to them the calyces. The calyces are what urine is going to first be obtained from when we get this out of the collecting system. And I really hope you guys can't hear my dog barking upstairs. I know I have noise canceling headphones on, but I don't know how strong they are. So the one of the things we're going to do with the minor calyces is we get the urine from the collecting system, and they will deliver that urine to the major calyces. The major calyces, they are collecting from the minor calyces, and they deliver urine to the renal pelvis. Now, the renal pelvis is, in this case, it's more like a funnel. It's the funnel that collects urine from all the major calyces, and it sends it down to the ureter. And the ureters is that tube we saw previously that went from kidney down to the urinary bladder. Now, the hilum of the kidney. The hilum, remember we talked about a hilum. We said that the hilum is a structure where you will have tubes, blood vessels, nerves, and structures like that attached. Like there's an indentation. We talk about the hilum of the lung, the hilum of the spleen, the hilum of the lymph node. Now we're talking about the hilum of the kidney. This is a word, a thing that we use many times. This indentation, you will find the ureter. You're going to find the renal artery and vein, the renal nerves attached here as well. Okay. Now, what we're going to do is diagrammatically, you can see those structures here. But what we're going to do is we are going to use a model to discuss these. And I'm going to turn to this model and kind of go back and forth on that. And here we have the model that I like to use. And one of the nice things about it is I can go back and forth here, and I'm going to go ahead and close this because I don't need that. Okay, so what we're going to do, we're going to talk about the renal capsule. Now, the renal capsule, remember, is external. It's this brown stuff on the outside of the model. You can see it all the way around here. You can see it here as well, the renal capsule. Now, the capsule being that thin external layer, next we have the cortex. The cortex basically is everything you're going to see right here up until you get to these little pink spots is all this is our cortex. Now, the cortex, then we're going to enter into the medulla. And the medulla, we said, is that inner lining. Inner layer is the medulla. Now, in the medulla, you have these things here called renal pyramids. Renal pyramids. Now, one of the reasons I always do this is so we can also see the word again. Now, we talk about renal papilla. We said renal papilla were the tip ends of the different renal pyramids, was the tips. Okay? Now, also, we talked about a renal column. That is between adjacent pyramids. So here we have a good example of a renal column right here. Between it, this is the best one on this model that we have, this band of tissue between these two adjacent pyramids. Now, the other thing we want to talk about is a lobe. A renal lobe, let's say that I've got this renal pyramid here. You have its overlying cortex, half of the renal column on one side, half of the renal column on the other side. And it would be this area that we would have, and you could kind of pull out. There is a very good diagram here that also depicts that a renal lobe is. Okay. Now, as well, we're going to see the sinuses are all the internal cavities inside. We can't really just point to that, but you can see there's various internal spaces, hollow areas that have these guys here, like your calyces in it, and things like that. Now, the other thing we want to talk about is the minor and the major calyces. This is where I like to use is here we have a minor calyx collecting urine from the renal papilla, and here's another. When you have these two minor calyces unite, you will form what is called a major calyx. This is actually a major calyx from the, like, coming from that side, like facing backwards, will be draining in. But this is your calyces. These are your minors. Together we have our majors, and that goes into our renal pelvis, which takes us to the ureter. Okay. Now, one of the things we want to talk about is the hilum, and the hilum, remember, is simply put this indentation here where you have blood vessels, nerves, and the ureter attached is the hilum. All right. Now, we will continue using this model because there's three things on it that I like to use. So then we want to go into the blood vessels. Now we're going to talk about some blood vessels. We'll talk about them. I hope you identify all the ones that we can find, and then we're going to go into the blood flow through these vessels. So we'll talk about the renal artery and vein first, then we'll go into the segmental arteries. Now, there are segmental veins, and they do exist. I know because I've seen multiple textbooks that do have them. For some reason, our textbook that we use does not include them. They don't give a great reason for that, and I have asked a nephrologist as well. The thing I'm going to say is they are not in there. They're not on our structure sheets. There are structure sheets. If you're taking my course for a grade here, then you know that what's on the structure sheets that are posted on our on D2L is what you have to know, and they're not on there for that reason. They do exist though. Now, the interlobar arteries and veins, arcuate arteries and veins, cortical radiate arteries and veins. Now, pay attention here. Cortical radiate arteries and veins are also called interlobular, not interlobar. Interlobular arteries and veins. Now, I can tell you that when you see extra names, I do expect my students to know those, and there's a variety of reasons. Remember, you may be working in the medical field. Well, somebody there has been working for 20 years, and they are 30 years in the medical field versus somebody who's been there 10 years. They may have very different educational backgrounds. Somebody may be hearing the more new terms. And interlobular is one that's not used much because it is easy to confuse interlobular for interlobar. But I do expect, and I can tell you that sometimes on my exams, I will go in and put interlobular instead of cortical radiate sometimes as an answer choice. The afferent arterioles, the glomerulus, the efferent arterioles, the peritubular capillaries, and the venules that are used. Now, this is not the vascular pathway. These are just major vessels. So let's start with the renal artery and vein. Now, the renal artery and vein, renal artery will be red in this case. Remember, the arteries are red when they are in systemic circulation. Kidney is part of body system. Now, with exception of pulmonary circulation and fetal circulation, systemic arteries are red. This is a renal artery, and the blue one down here is a renal vein. Next, we can turn our attention to the segmental arteries. Now, these are segmental arteries on this model. Now, of course, that would be a segmental vein. Segmental arteries until they branch. This is a segmental artery. Here we have segmental branches may come off. These segmental arteries branch off, and then they'll branch. And that's where our next branches are interlobar arteries and veins. Now, here we have coming off a segmental artery is an interlobar artery and vein because they are going there with the lobe between two renal pyramids, interlobar artery, interlobar vein. Now, next we're going to turn our attention and we're going to see the arcuate arteries and veins. Now, arc, that means they arch, they form an arc. The arcuate arteries, arcing here, and all of these are arcuate. You can see them arcing over the renal pyramids. Arcuate arteries and veins, and this is the only arcuate vein on this model. Now, as well, are cortical radiate arteries and veins. All of these are cortical radiate or interlobular arteries going out into the cortex, into the renal cortex. There is only one interlobular or cortical radiate vein on this model right here. The cortical radiate into the renal cortex, and that's where they get their name. Now, the afferent arterioles, glomerulus, efferent arterioles, and peritubular capillaries won't be seen just yet. I will talk about them later, as well as venules. They can't really be seen too well here. Um, there are some of them that are visible a little bit. For example, coming off, you can actually see a little bit of these right here on these models. Here we have our arcuate artery, our cortical radiate artery, and the afferent arterioles coming out to the glomeruli here. Um, and you can at least see those, but there are a lot of things that aren't visible on that model, but I am going to talk about them momentarily. Now, a big thing that I want to make sure that I teach and help you understand is the renal vascular pathway. Now, my students one semester came up with the mnemonic. Now, it's a little bit more older because most of you guys aren't familiar with that show, Ren and Stimpy. Actually, it was an older student of mine, one who's more my age, but that was a very popular show. Ren and Stimpy was somewhat kind of like the, um, um, I would kind of say the SpongeBob of my generation. Um, so, uh, but very weird cartoon. So we had Ren and Stimpy in a cage play very cool animations in reality. It was an animated show, so that was and cool was a word that was used back then a lot. So we're going to talk about the renal artery. We're going to start with renal artery. Then we're going to go to segmental arteries, ren and renal artery, Stimpy, segmental artery, interlobar artery, arcuate artery, cage. The C of cage is cortical radiate artery. The A in cage is afferent arterial. The G in cage is glomerulus. And the E in cage is efferent arterial. So Ren and Stimpy in a cage. Now we are out of our glomerulus. In our efferent arterioles, we have the peritubular capillaries. V, very, venules. C and cool, cortical radiate veins. Animations, the A, arcuate veins. N, interlobar veins. Reality, the R in reality is renal vein. And this is the renal vascular pathway. And you can see if we start renal artery to segmentals, to interlobar, to arcuate, to cortical radiate, to afferent arterioles, takes us to the glomerulus, to the efferent arterioles, out the peritubular capillaries, to the venules, to the cortical radiate, to the arcuate, interlobar veins, and renal vein. And we take this loop, okay? Ren and Stimpy in a cage play very cool animations in reality. Now, you can come up with your own mnemonic, and I usually recommend that you do because sometimes they're more meaningful to you. Not everybody is very good at doing that. What I sometimes do is, so I always give the ones that I've made or that a student has made. It was actually the last summer semester that I got to teach in person. This is our second COVID summer. So I have not taught in person now, and so it's been a while, and at least A&P labs anyway. I am teaching general and introduction biology courses physically, anatomy and physiology, since we don't, because of the models, we don't want them to handle those right now. But this fall we're coming back. So anyway, this actually came from that last one, and the student who gave me that is a nurse now. So Uh, she was finishing it off. Well, no, she's on nurse now, unless she went to our program. She is in nursing school now. Sorry, I apologize. So, all right. Now, here you can actually see all the blood vessels. Here is a diagram I wanted to use a little bit. We talked about the efferent arteriole went to peritubular capillaries. They are capillaries that wrap around the renal tubule. And there was a good chance to see that. Now, we are going to discuss the nephron. And one of the big things I'm going to do before we even go to do the nephron in like person as we discuss it and draw it out, before we actually look at the models, we're going to draw it out first thing. But let's talk about its parts, and then we're going to draw it, and then we will discuss some other things. Now, what I want to start off with is the nephron itself is made up of these three major compositions: the renal corpuscle, the renal tubule, and the collecting system. Now, on the side of this, we will see the juxtaglomerular complex, and we'll discuss that. Very important thing. But let's talk first about the renal corpuscle. Corpuscle here is discussing the glomerulus, which is the capillary bed, very specialized, about 50 intertwined capillaries is the glomerulus. And then the glomerular capsule, also called the Bowman's capsule. Now, from me, you are going to hear me usually call things first by the descriptive anatomical term before I ever use an anatomist's term. It was named after someone, Bowman. Now, the glomerular capsule and the glomerulus come together, make the renal corpuscle. The renal tubule, which is going to have a proximal convoluted tubule, the nephron loop, which is composed of an ascending and descending limb, and the distal convoluted tubule. What does it mean to be convoluted? It's going to mean that it is a little bit like squiggly, a little folded, more complex looking than not. It's not going to be straight. It's going to be a little convoluted. If something was like if you had like a road that was a straight shot, that would be non-convoluted. Collecting system, we're going to see it's made up of our collecting duct and the papillary duct. Now, as we go into this, we are going to discuss it, and sorry, I keep clicking the wrong thing here. Okay, so what we want to do is talk about the nephron. Now, with our nephron, now if you have me for class, you need to draw this somehow and post it onto our page so I can grade it. It is a drawing that I want you guys to do for me. Now, we're going to see it's made up of a renal corpuscle. It's made of a nephron loop. And it's made of the collecting system. Okay. Now, as we go into that, what I am going to do to start this off, and hard to tell sometimes what color I grabbed. Okay, so I'm actually going to start off with the glomerulus itself. Now, when I draw it out, what I like to do is draw the afferent arteriole as well as the efferent arteriole as well. I don't just draw the one thing. So what we're going to do is we're going to draw it like this, and we're going to have our afferent arteriole that is going to arrive. Let me erase that and fix that. Hate it sometimes when it's so cool down here where I am ink doesn't dry very quick. The afferent arteriole and the efferent arteriole. Okay, and these two here are going to be seen. So that's about these. And now let's also talk about the glomerulus. Glomerulus. Think about globe. It's like a ball, like a twisted ball of cells. It's globular. Think about it like that. It's this mass, a twine of, like a ball of twine. Now, this is just the glomerulus. Now, we have to make a renal corpuscle. We need something around this. We needed the glomerular capsule. We need our glomerular capsule. And together with the glomerular capsule or Bowman's capsule and the glomerulus, we have a renal corpuscle. And what this thing is going to do is enter into the area here called like our, well, I actually said nephron tube. What I'll just say is renal tubule here. I'll do that. Sometimes, I mean, the loop is there, but I can make that a little bit easier to follow if I do it that way. And I'm too far in to just delete and start over. So I'm going to call this the renal tubule. Just because I want to follow along more with what I was talking about there. So we have this set of tubes here. And what we're going to do with this guy is we're going to take part of this down. Okay, and then we're going to go part of this up into our collecting system, and we're going to draw that. Okay, so this is our nephron drawing. So what I wanted to do, and the reason I kind of changed this, we have our renal tubule, remember, has what's called a proximal, because it is closest to the glomerular capsule, proximal convoluted tubule. Let me write that a little bit over to the side there. I'm bad to paint myself in corners when I draw with less of a space, and I usually tend to paint myself in corners. Is called the proximal convoluted tubule, sometimes called the PCT. Now, as well, we have a distal convoluted tubule. It's more distant. Distal convoluted tubule, furthest from the point of attachment. Now, in the middle here, we have what is called a nephron loop. And this is why I kind of changed it last second. Our nephron loop is made of two distinctive limbs. There is one limb where the fluid that's in this is going down, and another limb where the fluid in here is going up. So you have what is called a descending limb, and then you have an ascending limb. Ascending limb. Sorry, I put in an E there by mistake. Ascending limb. Okay, now these two limbs are going to create our loop called the nephron loop or loop of Henle. And this is taking us to our distal convoluted tubule. Now, if we kind of imagine that the way this thing works, like this, our collecting system has what is called a collecting duct, which is receiving this fluid called filtrate from all of these distal convoluted tubules. And then in this area, you have this thing here. This is the renal papilla. So this is called the papillary duct. Papillary duct, okay? And this is our nephron. And I do apologize that I had to kind of alter my writing up here. It's sometimes when I'm going to draw it for teaching, it's not like when I make an official like video where I have a script and I follow my script. I'm drawing it more extemporaneously here. And I just want to make sure we had that. So this is the drawing my students would need to do. Remember, when you get this, what you're going to do is on the lab final, you're going to go into assignments, and there will be a tab called the nephron drawing for the lab replacement points. And we didn't end up doing this one. I left it, but we didn't end up using this one. I did something different. It was one for another semester. It's still in there, but we did something a little different this semester. Okay, so we drew this out. So what we're going to now do is take a look at this in a model. And I've got my model. Let me grab it and get it over here again and talk about it. When I use these models, we have some really good ones that I like to use. And of course, it's so let me see if I can get this kind of zoomed back out again. And also we'll need to just adjust this a smidge. Okay, so there we go. We're good. Now, this is the section here we're going to focus on. And you can actually see 1, 2, 3 nephrons here, and a part of one here. Now, we have cortical nephrons and juxtamedullary nephrons that are being shown here, but what I want to help you guys see is our nephron's basic structure. Now, they have it kind of crisscrossing here, but we're going to start here with the Bowman's capsule or the glomerular capsule, which means we have a proximal convoluted tubule here, which is, remember, highly folded. Then we go into a descending limb of the loop of Henle, and then the ascending limb of loop of Henle. Then we're going to go into the distal convoluted tubule in this light brown, and then we're going to empty into the collecting duct. Now, the collecting duct ends at the bottom down here, and you would have what's called papillary collecting duct that would end down here at the renal papilla. Okay. Now, model-wise, if you can draw out that sketch that we've done together, this drawing, then it is going to be a lot easier to kind of get these together. Now, the reason I separate this, it makes the physiology easier. I could tell you now, what I like to do with my students is, because things that we do to make sure they understand it. So a student of mine who throughout my classes, and she was studying for her entrance exam to nursing school. Remember, the reason why the entrance exams are done in these professional programs is not all A's are A's. You may make an A with a really superficial professor and not have the knowledge and cannot pass the entrance exam. Well, the student said this was on there. She said renal vascular pathway was on there. Ren and Stimpy in a cage play very cool animations in reality. The nephron was on there. And it's things that you need to know and understand. I don't just do it because I'm like, oh, let me show you how smart I think I am. It's more so that I can prepare you, because if you aren't prepared when you leave, you get in trouble. Now, one of the things we want to talk about is called the juxtaglomerular complex. Now, with this, the juxtaglomerular complex is, I'll see if I need to actually turn this light on here or not. I may, let's just see if this makes it a little bit better. There we go. It looks good. It's only half power. What I want to do is talk about our glomerulus we've mentioned right here. You can see it very well. We talked about an afferent arteriole and an efferent arteriole. Now, how do I know this is afferent? You will notice this one here is bigger than this one here. So the afferent and the efferent arteriole, and here we have the glomerulus, and you have your glomerular capsule around it. You can actually see here is the proximal convoluted tubule. Well, over here is the distal. The distal convoluted tubule in reality will come back and fold on here. It may be all the way over here, but in reality, this portion here would fold down. And this creates a very important structure for regulation of our blood pressures. And what that is called the juxtaglomerular complex or apparatus, sometimes called the JGA. Now, right here in these little cells right here that look a little bit more like a cuboid cell, these are cells called macula densa. Nearby here, there are cells called juxtaglomerular cells, specialized smooth muscle cells. Now, these cells talk to each other. Basically, what's going to happen is as blood flows into this structure, the stretching and things like that, and the chemoceptive nature, you're going to see that sodium and blood pressure is low. And then as the filtrate is produced and it comes back around out here, these cells are going to talk to these cells and say, yeah, volume in here is not very much. And together, these are going to produce the enzyme renin. They act as a mechanism to help to adjust the secretion of renin due to a decrease in blood flow pressure and sodium, and also a decrease in the production of filtrate coming through here. And this acts to talk to each other. So you have the juxtaglomerular complex. Now, where does it get its name? Juxtaposition. The word juxtaposition basically can say that I have two things that are side by side. Let's say that I'm at a fancy art exhibit. And at my fancy art exhibit, there is a photograph of somebody eating loads of hot dogs. And then next to that person eating loads of hot dogs is a painting of a starving child. And you looked at these two things side by side, you would notice that there was a juxtaposition of these two images. And that might be part of the exhibit to say this is a powerful message. You have this person just eating hot dogs, and then you have this starving child. And side by side, this makes a massive statement. Well, these are side by side. They are juxtaposed. Macula densa of the distal convoluted tubule and juxtaglomerular cells of the afferent arteriole. When you hear juxta, you're going to think they're in juxtaposition. They are juxtaposed. Okay. Here is a great diagram that also, this is a little bit like mine. Mine is a little bit more straightened out version of this, but you could see what I was doing here as well. Now, if you draw yours just like mine, that's fine. Remember, my students, if you just basically wanted to recreate this drawing, and there are some who are not really good at drawing sometimes, but the way that I drew it, you can draw it. But when you put pen to paper, you are thinking about it in a way that's not like what you always do. A lot of people are just thinking, oh, all I really need to do is just sit here and look at it. I was like, well, no, it's not just the way it always works. I will tell you there's special ways to do these things. Now, here you can actually see the renal corpuscle. Really good image here showing macula densa. And these cells here called the juxtaglomerular cells. Now, when I was a student, we thought that these cells here called mesangial cells made renin. We now know it's the juxtaglomerular cells that make renin. But we thought that's what made renin. Turns out it's not. So I always like to kind of make that point too, because it's like, hey, listen, we all have to keep learning. There's never just you're done. You're not just learning is something you never finish. You're never finished with learning. Okay. Now, in lecture, we'll talk more about this guy. We'll talk about the podocytes and their pedicels and the filtration membrane. But that is the structure there. Now, ureters. We don't spend too much time on ureters in terms of histology here. I do discuss their histology a little bit just to kind of help you as you guys get in if you deal with like antibacterial agents and things like that. I'm going to use this model here. I've got a model that I like to use to show all the structures again. And I told you I'd go back over it, and it is a rather large model. Sorry, went the wrong direction there. Very hard to get the whole thing under the camera, but we can get pretty much all of it this way. And we can see the two kidneys and the two ureters. Now, these ureters, what we're going to notice about the ureters is they travel down. They come to the backside, posterior surface of the bladder. Now, in this case, this bladder is from a male. How do we know? I see a prostate. I see the distal portion of the vas deferens and seminal vesicles on this model. This is a male. We'll talk about this in a moment. Now, with the ureter, what we're going to do is you can see those pretty well in this model. Now, I am going to talk about the urinary bladder. Now, the urinary bladder has these three openings, two entrances and an exit. There are two entrances on the top called the urethral openings, where the two ureters come into the posterior or back surface of the bladder. And they're here, and you can see them pretty well here. And then at the bottom, there is an opening called the internal urethral orifice, the entrance to the urethra. And that makes 1, 2, 3 holes, which makes a triangular shape called the trigone. The ureter openings and the urethral opening called the trigone. Now, at the apex of the trigone, at the end of the trigone, there is where you would enter into the urethra. Now, this is a male. Again, prostate is found in males only. Now, the rugae, remember rugae of the stomach. Rugae are folds in the mucosas of organs that need to expand. Now, typically you're talking about like 350 milliliters, and you got to pee. 500 milliliters, and you're going to basically force the bladder open unless there is some problem. I was discussing this with a class, and they told me, some of my students are at least they already have some professional job in healthcare, and now they're coming back. They might be a certain tech or something like that. And now they're going to get a more advanced degree and become a nurse. And if you are watching this in another country, in America, of course, this is nursing is quite a bit different because you can go all the way to a doctoral level program in the nursing realm. Some countries don't do that, but anyway, they were telling me that they actually had gone in to catheterize a patient. It was a male patient, and they got over 1,000 mils out of his bladder. I don't know how it didn't rupture. But the rugae allows for this expansion. Now, inside, we're going to see the detrusor muscle. These are the muscles that will actually squeeze to help empty the bladder. Now, what I want to do is to show the bladder a little bit better, we're going to detach our ureters, and we're going to take this off. Now, I think somebody was trying to do something demo with this. I've been noticing that there is oil all over these models, and I mean it's not gotten in the bladder because it's not all the way open. And I think maybe somebody was just trying to do a demonstration in class once, and I was like, I don't know, but anyway, we're going to take this guy off. I'm going to zoom in here. A very good model that we can use here. I might adjust the light a little bit. Sometimes it is better, sometimes it is worse, but I think that's a little bit better. There is some natural light. It is late afternoon when this is when I'm filming, as you can see. So, all right. As we talk about the urethral openings, now to find them first, this is where they came in from the external surface, here and here. So if I flip it around, I'm going to find those openings here and here. Now I might turn that light on. I'm going to turn it to this lower setting. This is kind of a little bit better. So we have one here and we have one here. Now this is the opening to the urethra. So this is called the trigone. And the rugae is this roughened mucosa structure. And then lastly, what we can see is our detrusor muscle. This red line represents our detrusor muscle. And the detrusor muscle is what squeezes the bladder to help empty that. So our urinary bladder there. Now, normally when I teach this in fall semester, this is the last thing we do before students leave for their Thanksgiving break. And then they come back and we have our last lab, and then we have our lab final, and they leave for lecture finals. So as we approach it, but when they go to Thanksgiving break, I would always put this up because to me it looks like a turkey, and I'd always have to wish them a happy Thanksgiving with a bladder for some silly reason. Um, it was always a thing that I just couldn't help myself. Always had to do that. Now, the urethra, there is actually an internal sphincter here in the neck of the bladder. And then down here, the external sphincter, which is actually the urogenital diaphragm, part of pelvic floor. Now, in males, there's this space where the prostate gland is. This is what prostate surrounds the urethra, why an enlarged prostate impinges it and makes it difficult to urinate. So, the things that you guys will make sure you know all the functions, all the components, what everything does. Be able to identify that. Basically, you got to know everything that's in here. One of the things I do have my students do is go through these, and these are the models that I could be using for the examination and the structures you would need to be able to identify all that for the examination. For here to practice on. This is since I went online, this is my way of basically doing lab. Now I had thought about it, but there's this people said, well, why don't you do stuff? Because I'd hear people discussing, well, so and so at this college has done this really big thing with like these interactive visual body things. Well, I do not, in the community college, do not want to cause my students extra things they have to pay for. Basically, if you have me, don't need really even. I provide a free textbook. All my notes are made because part of this, we want to keep costs low for students, but maintain a quality education. And that information being given is always good. So as you complete this, these are there are no actual diagrams that I will be using for this examination. It will be all models. And with that, we are done with this one. We have one more left, the reproductive system. Now, for those of you guys, it's already up. I had to redo a few labs and some lectures because of problems with transferring them, and we lose things. The system we use is called UJA. It's great. I love using it. It's just that it doesn't keep things for more than a year. And that's why I went and moved all of my stuff through COVID onto my YouTube channel. So guys, thank you so much. If you are watching this on YouTube, please feel free to give me a like and a subscribe. But all of my sins are, of course, now. But this means that anybody who takes who doesn't have me as a professor for this class in the future can still watch it, even if they have someone who does a very poor job in their class, making sure they don't show everything. So thank you so much. Have a great day, and we'll see you in the reproductive system for the very last lab. Thank you guys, and have a great day.